So welcome everybody to this second day, the second module of the course on multivariate approaches to soil microbial communities. My name is Corrado Costa, and today I'll talk you about multivariate analysis of variance MANOVA. Before we have to arrive to MANOVA, but I would like to introduce you the general questions on the analysis of variance. So I prefer to start from the univariate approach just to introduce you the basic concepts. So we can start introducing the different kind of indexes. So we have position indexes such as mean, mode, median, different kind of those kind of indexes, and dispersion indexes such as range, variance, standard deviation. Everybody knows this kind of indexes. This is just an example. Basing on the same mean, we have these three different kind of distributions. So this is a platycourtic distribution, so with a huge variability, a mean distribution, the green one, and a leptocourtic, so a distribution with a low variability. Today, all these 2 hours, we will talk about the variability, so that we call variance, but we will arrive there. Very short introduction on the interval of confidence, the 95 confidence intervals. So are based on the mean standard error, and the statistical meaning is that when we applied 100, for example, different analysis, 95 will be within the range of the true values, but not 5. So here is in the yellow part of this graph, we have the confidence interval. This 2.5 here and 2.5 there are the probability rejected, so outside the confidence intervals. In short, the interval is right 95 times, 95% times. Which is the null hypothesis and the alternative hypothesis in the difference between mean values. So the null hypothesis is that the means of the groups are all the same. The alternative hypothesis is that the means of the groups are not all the same. We will go in deep, very in deep with this. So if any one of you have questions, please stop me and ask. Basing on this, we have two types of errors. Type 1 errors, alpha error, that is when the null hypothesis is rejected. So the null hypothesis fails, but the real condition is that this is true. So this is a false positive. The type 2 error, the error beta, is when in the experimental condition, the null hypothesis is not rejected. So the null hypothesis is true, but the real condition show that the null hypothesis is fails. So we have false negative with different kind of confidence intervals. So the basic soil is 0.05 or 0.01 or 0.001. So different levels of significance. In general, when you read papers, I read a lot of papers on this kind of matter, so on statistical, but also on basic statistics. There are a lot of misunderstandings when the probability, what the probability of significance really is. So if we, for example, we observe that the probability P of difference between two means is P equal to 0.03 or 3%. The correct statement is there is a 3% of possibility of observing a difference between the means greater than that found in the data when the means of the population are equal. So just to understand this sentence, even if it is correct, but is very hard. In other cases, we can find also incorrect statements like the second, the B1. So the 96 probability exists that the difference between the means depends on the real one difference, and that is 3% the probability that it can only depend on the case. So apart the fact that this is not correct, both of them are very hard to be understandable. You need a couple of or 5 minutes to well understand what the author would like to do. So I suggest, this is my suggestion, because if we say something easier, it is much more understandable and it is easy to be transferred to a reader. So it's much more correct to say that the null hypothesis H0 that the means are equally is rejected or is approved. This is what we discuss all this afternoon. So the comparison between two sample means. So we will, I will show you the t-test or the one-way ANOVA, or later on the MANOVA, of course. And what is the basis of the calculation? Is the ratio between a signal and a noise. Well, what is signal and what is noise? The signal is what the investigator is trying to demonstrate. So the difference between group means. So let's do an example. We have two different cultivars of apples. We would like to assess if the means of the fruits are the same or not. So the signal is the weight, so the mean weight of cultivar 1 and the mean weight of cultivar 2. And the other side of this ratio is the noise, so the variability of the group. How much the apples are variable considering the weight. So this weight within the same cultivar could be large, or could be small. Smaller is the variability, higher is the probability that these two means are different. We will go very in deep with this argument, but this is the basis. If we well understand this concept, we have understand most of the lesson of today. So the first step, we will start from the easier design, and we will go with complicating the examples until arriving to the multivariate differences. That is quite more complicated. But why we would like to arrive to the multivariate analysis of variance? Because in general, in the multivariate statistics, it is hard to assess a significance. So it is a way of assessing a significance in a multivariate analysis. And so we will arrive there, but one step after another. So the definition. What is a t-test? Statistical test for the analysis of significance is based on the possible refutation of the null hypothesis at a given probability of difference between a mean and the reference value, or between two means. Comparison of the calculated index t with the limit obtained from the theoretical distribution of TD student. So we have to infer that two samples mean different differ significantly, so rejecting or not the null hypothesis of equality between the means. So for a given level of probability alpha, for example, 0.05 or 0.01, must result that t-value is higher than the critical values of distribution t of the probability. We have three basic assumption. The first one is most of all depending from the investigator, so the independence of the collection of the observation. So we have to take care of this, because if you get the apples, the reddish one, because it is much more visible than the greener one, it is not an independent collection. So you have to be sure that this independence is done. So the second one is the normality of the sample distribution. So the distributions should be normally distributed. There are different tests. We will see something about that very, very rapidly. And the third one is the homoscedasticity. This is a very bad word, but is very important for the t-test, but also for the ANOVA that we will see later. In general, I could say that the normality test and the homoscedasticity tests are not so often reported, even if scientific papers. But I suggest you to follow what this afternoon I will show, because in my opinion, it is very important to demonstrate the normality and the homoscedasticity. Because if one or the other are not verified, you could not apply a t-test or a test ANOVA, but there are different tests analog to t-test or ANOVA that could be conducted with the real, with the proper basis. So let's start with the normality test. The null hypothesis of the normality test is that sample taken from a population with normal distribution. So if P, between parentheses, that there is the null hypothesis, so normal, is lower than 0.05, the normal distribution can be rejected. So this distribution is not normal. Shapiro-Wilk is one of these tests and is considered to be the more exact, but of course it depends from the kind of data that you have. So we can start with a simple exercise. Yesterday I sent you the WeTransfer with the software where there are past that we used yesterday and also today. Now we will use past, but also the 50/50 MANOVA that you have to install on your PC for today. And today I sent you the WeTransfer with the material of this lesson. So within this WeTransfer, you can find the file ANOVA MANOVA exercises and the sheet. We go to the sheet normality. So ANOVA MANOVA exercises. Okay. This sheet normality here. Here we have three distributions with the one with the normal variance, the kurtosid one that is the red one is much more flat, so the variability is higher, and the skewed, so the asymmetrical one is the green one. We have to test if these three distributions are normally distributed. So we copy the We copy this data set, Control C. We go in the software, we open past, the past executable. The other file is the manual. We have to click on Edit Labels. We do not have rows labels here, so or we copy from here, from the base here, or we can copy just this part. In this case, we have to paste here in A, and again edit labels. We are ready to analyze, to study the normality of these three distributions. So as you remember from yesterday, we have to select the columns that we would like to analyze. So all of them, I click here. In the menu statistics, here we have the univariate statistics. We conduct a normality test. Where is it? Normality tests here. Okay. Let's open a bit more. So here we have the three different distributions, and the n is the number of samples here. This is the Shapiro-Wilks value of the statistics, and this is the p, the probability, where past helps us putting between parentheses the null hypothesis. So the null hypothesis is that we have a normal distribution. If this value of p is lower than 0.05 or 0.01, the null hypothesis of normality is rejected. In this case, the first situation is normal because p is higher than 0.05, and also the Kurtoset distribution, the red one here, is normal, but the skewed one is not normal, so the null hypothesis of normality is rejected. Okay, everything is fine? I hope so. Past reported other kind of normality test, but I suggest you to use the first one, so the Shapiro-Wilk and P relative to Shapiro-Wilk. Okay. In this case, we could analyze the first and the second distribution, for example, for a t-test, because both of them are normally distributed, but not the skewed distribution. Okay, let's go on. We have different kind of t-tests. So we have one sample t-test. This test is used to verify the null hypothesis that the population mean is comparable with a specific value. Let's do an example. For example, the mean milk production of a cow Okay. And if the national milk production is, don't know, 100 liters, I say a number, just a number, and I would like to know if my production of milk is comparable with the one of the mean Italian production. I compare my distribution of my cows with the single value of the Italian production, so that 100. I have another possibility, so independent two-sample t-test. This is the most common case. So we have two columns of the same variables, two columns with two different, don't know, such as the apples weight. So two different cultivars, 100 apples for each cultivars, and we would like to test if the means are or not the same or otherwise comparable. So if the distribution are homoscedastic, we apply a t-test homoscedastic. Is the distribution are heteroscedastic, we will see this within a few minutes, we will apply the Welch t-test. This is the second option. The third option is if I have only the parameters. So if I have only the mean, the standard deviation, and the number of samples, I can apply this third option. The last case is the t-test for paired samples. What does it mean? So paired samples means that samples are said to be paired if there is a dependence between factors level. For example, if we have two cultivars and we have of apples and we have 100 apples for each cultivar, if we mix the number of the apples within the same cultivar, it doesn't matter. The result is the same. But for example, if I measure something on a cow or on a patient before or later that I gave medicine or something, and I mix this kind of information of different cows, the results change. So paired samples is when you could not mix the samples within the same column. Let's start with the t-test from parameters. Okay, so let's go in past. With the parameters, we don't need real data here. We go to statistics, F and T from parameters. And here we have to put the values of mean, variance, and number of samples. So let's start, for example, with the example of the apple, the weight of the apple. So from one side, 50 grams. From the other one, 60. Okay. The variance is 0.12 here and 0.24 here. The number is 100 and 100, so the same number. We can compute here. And first of all, there is the homoscedasticity test. So the null hypothesis is that the two distributions have the same variance. The null hypothesis is rejected because the values is lower than 0.05. So in this case, we have to look at the unequal variance T, the Welch unequal variance T. In this case, the null hypothesis is the same mean, and here is highly significant, so that it means that the null hypothesis is highly rejected. So let's try with changing some values here. So we try with the 13, leaving the same here, compute. The f-test become non-significant. That means that I have to look at this row, the t-test, the homoscedastic test, t-test. The null hypothesis is the same mean, and of course it is highly rejected. Let's try to increase the variance, so 0.8 here and 0.83 here. We are just playing with this tool just to observe what does it mean. Compute. It is at least let's increase drastically with 2 variance 2 here and 5 5 here. No, it's okay. 15 and 15.3. Okay, it's lowering. 52 and 53 here, the variance. Okay, much more. Okay. Okay. Okay, we are approaching to the same mean. So I am increasing the variance, so the distribution are much more have a higher variability, so have a higher variance that cover the effect of the two different means. So I need a very high variance to cover this effect. So with 1,000 here and 1,100 here. So the F-test is not significant, and the T-test is not significant too. No, is at least significant. So we have to. Okay, let's try now. Okay, now they are not significant. So the two means, even if equal at the other trial that we tried, become non-significant because the variance increase. So let's reduce the variance to 100 here and 110 here, and reduce the mean in the second sample. So 55 is at least different. 52 is no more different. But if I increase the number of samples to 1,000, here, it becomes significant. This is very, very important for all of you that are investigators. That means that when a statistician tells you to collect much more samples as possible, is because it is important to have a more heavy possibility to arrive to a significant value. So you see that with the same number, just reducing the number of samples, it is not significant. Increasing, it becomes significant. Okay. Any question on this? Okay. How many we are? 17. Okay, less than yesterday. Okay, let's continue with our lesson. So let's try with the t-test, the option here, the t-test with 2 samples. We have to go in the file MANOVA exercises, the sheet t-test here. Okay, where is the? I couldn't find the, no, sorry. Exercises. Otherwise, we can get these 2 columns here. So One Way Ortofrutticoli, Romana and Trocadero. We can copy this. File, new. Okay. Edit labels. We copy in A here. Paste. Edit labels. We select both columns here. And we can go to Statistics, F and T-test two samples. So you click on the first, move on the second, shift and click on the second. Statistics, F and T test, two samples. Okay. So let's see. We have here the description, the basic statistics for the first distribution and the second distribution. Okay. First of all, we have, of course, to test the normality. The normality test confirm the null hypothesis of normality. The values here are higher than 0.05. Okay, so I can continue with the FNT tests. So we have the mean of the two distributions, the 95 percentage in confidence intervals, the variance here. Okay, and we have the F test, the homoscedasticity test, that confirm the null hypothesis of same variance. So I have to look at this row, t-test. That is the homoscedastic t-test. The null hypothesis is the same mean, and this null hypothesis is rejected because the value here is lower than 0.05. It is also lower than 0.01, so is highly significant. Okay, so let's go on. Okay, we have seen this. Now we have observed the example with the difference between two distributions. But if we have to compare more than two distributions, we have to apply the analysis of variance. It used to compare internal variabilities of this group with the variability between the groups. So the analysis of variance used the variance to observe the difference of means. We have, as we said before, two kind of variation. The one systematic under control of the investigator, differences of weight of the two cultivars, of the three cultivars, and the random source of variation that is under the biological variability, the environmental conditions. So signal versus noise. And we have to observe if is prevalent the systematic variation or the random variation. This is the game. The null hypothesis is similar to the t-test one, so it is assumed that the mean of all the groups are equal, and the alternative, it is at least one group mean is different from the other. The assumptions are similar, of course. There is the independence of the samplings, the normality that all distributions are normal, all distributions, the homoscedasticity. Data from all groups have a stochastic distribution and comparable variability. So very similar to the previous one. MANOVA is used to compare more than two means. Consider more than one factor or predictor. We will go in deep with this definition. Despite the name, MANOVA does not compare variance, but use variance to compare means. Tell us if the means of groups are different. It does not tell us exactly which mean differs. For this reason, the post-hoc tests have been developed. Some terminology, it is important. What is a factor? A factor is the variable used to differentiate a group from another group. That means the cultivar in the cultivars in the example of the apples is a factor. So cultivars is the factor. The level of the factor is the possible values, states or characteristics that the factors can assume the cultivar 1, cultivar 2, cultivar 3. The factor is cultivar, the level is 1, 2, 3. And the response variable is the quantitative variable under the study. So in this case, the weight of the apples. Okay. We could have different experimental designs. So if we have just one factor, it is to say that we use a one-way ANOVA. So one factor, single factor, one-way ANOVA. Two or more factor, two-way ANOVA, three-way ANOVA, or in general, multi-factor ANOVA. The treatments are determined by combination of levels of multiple experimental factors. Each treatment is applied to one or more experimental units. Of course, here we have to find the differences between means in different groups. This is the basic. Here is an example of the analytics. So we have the level of factors, 1, 2, 3, etc., and the replicates from 1 to n. Now, maybe for the first and the last time in your life, we will go in deep the procedure of an ANOVA. I believe that this is important because looking at the calculation to extract an ANOVA serves as not only an exercise, but to well understand what an ANOVA do, and for an investigator, how to construct your experiment in order to be well adapted to the case study that you want to operate. So this is the example. So the file is the same and the sheet is one-way orthofructicoli. So here, this is the data set. We have 1, 2, 3, 4, 5, 6, 7 different horticultural species and 10 repetitions. So 10 measures of the same variable, that is, I can't remember, is a colorimetric coordinates L of the CHILAB space. So is a color measure. So we go to past, file, new, okay, edit labels, click in A and paste, no. Copy all of this and paste here. Okay, and again, edit labels. This is the data set that we will analyze. So, first of all, in my opinion, it is important to observe this data set. One way of observing this data set is, I suggest, a graphical way of do this. So we select all of these on plot. It is possible also in Excel from 1 year. So we can apply bar chart box plot. So the box plot is now present also in in Excel. So box plot here. And we can observe the distribution. So a box plot is made from a mean value, that is this line here, a box that in this case is the confidence interval. No, it is the quartiles, and the whiskers here. So the name is box and whisker plot, means the 95 in confidence interval here. Okay, so we can observe that Romana is comparable in a part with Susina, but not with all the other. This Trocadero is comparable with Susina too. These two, Nettarina and Pesca, are very, very similar, but also with Zucchina here. And Pomodorino is similar only with Zucchina. This is the general visualization. Okay, we start with statistics normality tests, and we observe that all the distribution are normal. So the null hypothesis of normality is accepted. So we can go ahead with the ANOVA and go on. Okay. Okay, we paste all of these. Normality test, we did this. So the inference, so the null hypothesis is that all the means are similar. The alternative is at least one of the distribution are different, have a mean non-comparable, significantly different from the other. Okay. The basic brick of the analysis of variance is the deviance, that is named sum of squares, is equal to the sum of the square of the deviations of n values, y, from their mean y mean. Okay, this is the basic brick to build this wall. We have the decomposition, so we have the variability within the groups, experimental error, and the variability between groups, the treatment effect, that is the effect that we would like to study. So we have a total deviance, total sum of squares, a deviance within groups, sum of square or error within groups, a deviance between groups, sum of square between groups. And this is the total with the two components, treatment effect and experimental error. So the total is equal to the sum of the within plus the external one. So the null hypothesis is true when SSG is relatively small, so that SSE is much larger than SSG. The alternative, if the null hypothesis is false, SSG should be relatively large. Therefore, SSG is much greater than SSE. Graphically, what is the sum of square total, the total sum of square? So if this is the overall mean of these three groups, is the square distance of all observation from the overall mean. This is the formula. This is the calculation, and this is the total value. So for each species here, I can calculate those values, and this is the sum for each group. So the sum of all these squared distances from the mean. So this is the sum from this, the sum from this, and the sum from this. And this is the numbers, and this is the total sum. What is graphically the sum of squares within groups or error? It is the distance, the squared distance from the mean of each group. So we calculate this distance from each observation within each group from the mean of each group. This is the formula, This is the calculation, and this is the total sum for each group. What about the sum of squares between groups? This could be quite difficult to be graphically understandable. However, you have to figure out that you have an overall mean, that is the one that we observed with the total sum here, the blue dashed line, and the mean of each group. That is the SSE that we said before. And we used to consider as a single observation as the mean of each group. So it's the distance of the mean of each group from the overall mean. So here you can see that the values are always the same. This is the sum for each group and the grand sum. This is the number. So SST, 9,000, etc., is equal to these two values. It is demonstrated. But if the null hypothesis is false, is rejected, it means that SSG should be lower than SSE. And this is so. So SSG 7,000 is lower than SSE 2000s. Okay. Let's go ahead. So we have to introduce the degrees of freedom. Why? Because the degrees of freedom we have three kinds of information. So the total elements, the elements of the group, and the elements per group, the elements, the number of groups and the number of elements per group. So the deviance, the total deviance, SST, have M total groups less 1 degrees of freedom. The deviance between groups has P groups less 1, that are the degrees of freedom for the SSG. And for the SSE is M minus P degrees of freedom. Similar to the computation, the degrees of freedom, the total is equal to the sum of the other two. Okay, so the variance, the formula of the variance is the sum of squares divided by the degrees of freedom. So we have the total variance is the total deviance divided by the degrees of freedom total. So M minus 1. The variance between groups, the deviance between groups divided per the degrees of freedom between groups, etc. So, but be careful that the variance, the total variance, is different from the sum of the two, of the other two. Here I show you a table where in the different articles you can find different terminologies for the thing that we are showing. So in the sheet MANOVA terminologia, you can find the different kind of names that you can find for the thing that we are saying today. Okay. We have to use the F-test, that, if you remember, is the same test that we approached to test the homoscedasticity in the t-test, before the t-test. So the t-test analyzed difference in variability, in variance. We have F, we have a variance within groups, between groups versus variables within groups. Signal against noise. This is the values of the of the test. This is the critical values, and the probability is this one. So the ANOVA test, F test, showed that the null hypothesis of comparability of the means is rejected. So at least one of the seven species are different from the other. But how could we? No, first of all, we try to do this in past. So we had these 7 distributions. We select all. We go in statistics. One way ANOVA here. Okay. Now we understand what does it mean. Maybe many of you used an ANOVA and copy from the tables the sum of squares, degrees of freedom, mean square, F values. But now I believe that all of you are able to understand what does it mean. So between groups, we have this value. Within groups, we have this value. This is the degrees of freedom. So we can calculate the mean square here, and this is the value F. It does not report the critical values, but the P statistics show that it is significant. So the same mean is rejected. To understand why this is rejected, we have to look at the post-hoc test. First of all, unfortunately, past is not well organized because we have tested the normality before. Then we have to test the homoscedasticity. So this row should be positioned above. So Levene test for homogeneity of variance. This is confirmed. So they have the same variance. And so we can go here to the F test with the same variance. Otherwise, if this test has been rejected, we have to go there, simply there. It is the alternative Welch F test in the case of unequal variance. And however, this is the value. Both of them are rejected. Here we have the post-hoc test, in particular, past reported as the Takei pairwise comparisons, where in this part of the table, you can find the Q values of the Takei test, and in the upper part of the matrix, you find the P value. So this is paired comparisons. So you have to look where crossing the rows with the columns, where they are significant. In particular, past helps us coloring in pink the values significant. So between Romana and Trocadero, I can show you also the okay. So from Romana and Trocadero, there's differences. They do not overlap from the box part, so they are different, but also from Nettarina, Pesca, Pomodorino, but not with Susina. They are similar, comparable here. In fact, Susina are similar. Trocadero is different from all apart the Susina also. This is true. This is true. Of course, this is true, but this is also graphically observable. For any question, I'm here. Nettarina is quite completely equal to Pesca, 0.9998, so is quite overlapping. And also with respect to Zucchina is comparable, and also nettarina, pesca with zucchina is the same, and also pomodorino with zucchina. Pomodorino and zucchina is the same, while the other pink are different. Okay, so just to confirm. Any questions? No. Okay. Let's go ahead with the presentation. No, just another thing here. I would like to show you also the case of repeated measurements. So when in the case that the measures are repeated, so we have different timing in measuring something in the blood after giving a medicine to different patients. So in that cases, I click here, and here you can observe the values and interpret in the same way, of course, with different results, because the statistics is quite different. Okay. We have observed the post-hoc test, so paired comparisons. Just a few words on the Bonferroni correction. Bonferroni correction is always used when you have many comparisons and few observations, such as in that case. This is the formula to calculate this, but of course the softwares gives you the results with the Bonferroni correction, with or without often. Bonferroni correction is a sort of penalty that you give to the results of a pairway comparison, so that to better stress your results and to better demonstrate the effect of something. So beyond one-way ANOVA, you have more than one factor. In this example, we had just one factor, that is the species, and 7 levels, that is the name of the different species, so pomodorino, zucchini, etc. You can have a different factor design, so with 2 factors, two-way ANOVA, 3 factors, three-way ANOVA, or n factors. So this is factor 1, for example, different years, A, B, C. Factor 2 are the different cultivars. And the question is the weight of the 2 cultivars different, considering also the factor year? So the experimental design is quite more complicated. So the advantages of the factorial ANOVA is that allows a greater generalizability of the results, require fewer observation, is much more economic, and reduce the error variance within cells, so it is much more powerful. And it allows to highlight also the interactions between variables, to the joint effect of the independent variables on the dependent variable. So what is the interaction? The interaction is of course, the interaction between the effect of the 2 factors. So in general, you have to discuss, first of all, the interaction of the effect. If this interaction is significant, you can only discuss the interaction without observing the single factor. However, you can say few words also on the single factor effect. This is statistically, we do not go in deep as before, but we have the sum of squares total with the contribute of the error, so the noise, and the the sum of squares between. These sum of squares between have an effect of the interaction, an effect of factor A, an effect of factor B, etc., etc. This is the composition, the decomposition. In past, you have to put the data in a different way. So, two-way formaggio here. Here, okay. So we have in this experiment we have cheese with different months of maturity, 1, 2, 3, 4 months, and the can't remember which value is the pH of the cheese is measured in the external, mean, and internal part of each cheese form. This is the matrix, but to be imported in the past, you have to put the two factors, month position, numerically into columns, and the third column is the quantitative variable. Okay, this is the way of preparing your matrix. You copy this one with the header, copy. You go in past, file new, okay, edit labels. You paste from A here, edit labels. You get the three columns here. Click on the first header, move with the mouse on the third, shift, click on the third. In the statistics menu, you can find two-way ANOVA. Okay. This is the result. It is quite similar to the one that we have observed with the one-way ANOVA, but we have to remember to start from the interaction line. So first of all, we have to look the interaction and then the single factor effect. So let's see the interaction. So interaction is not significant. So considering the interaction, there are not significant effect of month with position, 1 month per position. We have to look at factor B. Factor B is the position, not significant, so no effect of the position of the pH within the cheese. And factor A instead is significant, so there is a significant effect of the month, so the maturity, so the maturation of the cheese. With the maturation of the cheese, the pH changes significantly. This is the reply to the question that we did. Quite easy. Okay. So now we can go ahead. Of course, also here we have repeated measures. And so if you have a repeated measure design, you can use in this way. So this is a very, very, very important slide. I call this slide the slide of tranquility because sometimes we do not make the normality or homoscedasticity tests because if they are not confirmed, we don't know what to do. Why? With this slide, there are the different options. So, for example, if the normality is verified, this is what we did today. normality is verified and homoscedasticity is not verified, we add the Welch one-way ANOVA or the alternative that we already observed. But if the normality is not confirmed, we go inside the other kind of tests, so the non-parametric tests. In the case of homoscedasticity verified, we have the Kruskal-Wallis test. In the case of homoscedasticity unverified, so rejected, so heteroscedasticity, we have the Friedman test. But, for example, in past, we have just a different row. So Friedman test and Kruskal-Wallis test. So after the normality test, we can, if it is not confirmed the normality, we can choose Kruskal-Wallis or Friedman. That's it. But what about the post-hoc test in the two-way ANOVA design? They are not reported because the two-way ANOVA is made for to observe the interaction. So this is the way of doing, and past is limited from this kind of information. So for more complicated experimental design, you have to skip to another software, of course. Now after around 1 hour, we arrived to talk about MANOVA. Is there any question? No. Okay. So we enter in the multivariate world. So the multivariate analysis of variance have all the basics that we introduced with the analysis of variance, but with the complication that we have not only one variable, so we are not in the univariate world, but we enter in the multivariate. So we will observe altogether more than one variable. It is possible to test the multivariate normality, and in the case that multivariate normality is not assessed, we have the non-parametric MANOVA. This is the alternative. Such as for the univariate case, we have an hotelling T when we have to compare two groups, a one-way MANOVA if we have one factor and more than two groups, or the factorial MANOVA if we have more than one factor. Okay. The null hypothesis also is similar. It's complicated because the mean is not a mean of a single values of a single variable, but is a grand mean considering the multivariate mean. So it's quite complicated to understand, but the tests show you these. You could apply these to the matrix. The null hypothesis is the same, that at least one of the groups is different. Hotelling T in past is, let's see if we have time for this. Just a moment. No, we don't have an example here. Okay. Okay. So if you have a multivariate data set and you want to test the significance of the difference between groups, you have to apply a MANOVA. I suggest I introduced you the 50/50 MANOVA. I gave you the software. You have to install this software. This software is very interesting because it is not a classic MANOVA, but this approach has been introduced by Langsrud on 2002 and developed a generalized multivariate ANOVA method named 50/50 MANOVA. That instead of testing single principal components by separate tests, one multivariate test for several PC is performed. I would say here is some the packages, the references. Okay. So let's try with the open the software I have here. Okay. And let's see. Let's try with this environment Excel. Okay. So few basic rules. The file should be an Excel file, but not XLSX, but the older XLS. So the older version. The version Excel 5.095. Okay. Okay. Then you have to put the name of the variables. And here you have the factors. Here, these 1, 2, 3, 4, 5, 6 factors. The crop code is similar to the one that we observed yesterday. So the crop code, the MAN method, the condition, the treatment, the partner code, and environmental zone. And here you have the variables. So this data set is based on environmental variable. Okay. This is a very complex experimental design. So we have many 1, 2, 3, 4, 5, 6 factors and 1, 2, 3, 4, 5 and 6 continuous variable, quantitative variables. Okay. So the factors should be expressed with numbers, continuous numbers, 1, 2, 3, here 1, 2, 1, 2, 1, 2, 1 up to 10, and here up to 6. Okay. And that's it. It's better if you have only one sheet inside the file. And this is the basic rules. So now we can start loading the file. You have to reach the file. Sorry, I just copy here the. Okay, so let's start with environment XLS. As you can see, he reported here the name of the first row of the variables. It shows you a part of this and tell you that you have 33 rows and 12 columns. In the first line is the column names. And that's it. Okay. Now we have to tell to the program the experimental design. So we have X and Y. Y are the quantitative variables, X are the factors. So columns, we have from 1 to 6 are the factors. So we select all of these, putting in the right side. Okay. So we have selected the first 6 here. Okay. And then from the other side, we have to select the other 6, that is the quantitative variables. Okay. As for yesterday, I suggest to standardize the variables, and then we have to decide the model. The model means which kind of factors and interactions between factors I have to analyze. So we have it is very complex because we have the single factor, the first 6, then the couple of factors, and then the effect at 3, at 4, at 5, and HOI in, I believe this is Swedish, means all. We can select all or decide just to observe the single factors without any interaction. Let's try. Okay, only the factors. Okay. And then simply run. As you can see, the result of a MANOVA is very similar to the one that we observed with the ANOVA. So the degrees of freedom, the sum of square variance, and we have also the number of PC, the number of principal components. This is different from ANOVA. And then you have also the variance, etc. But of course, this is much more important, the p-value. So we observe that the environmental variables, the multivariate environmental variables, is significantly depending from the environmental zone, from the partner code. So the partner code show a different, highly significantly different environmental conditions. The treatment is not significant. The condition and management are, and also crop code are significant, but very light significant. And you have performed the MANOVA. The advantage of the 50-50 MANOVA is that you can also assess Y. So which variable, which is the contribute of each variable in this in case of significance. So for example, we would like to observe which variable are more important for the partner code, for this partner code significance. And this is done by a rotation test. So we click on rotation test. We select which interaction, or in this case, which single factor we want to analyze. Partner code here. And this rotation test is made with an iterative procedure. So the simulation is conducted on how many? 100,000 trials run. He is working on this. He completed the simulation. And here you can observe in this column, in this last column, the contribute of the variables. So all of these are significantly giving their contributes, but more than the other, the first four. So precipitation seasonality, temperature seasonality, temperature annual range, and also mean diurnal range. And that's it. So you can observe also the contribute of the variables. This is very important, and not all the MANOVA software, the software that made MANOVA returns you also this information. Most of all, they stop here to show you the single factor and the interactions. So try with another data set, microflora NGS here. This is, of course, an XLS file. Okay. We have also in this case 1, 2, 3, 4, 5, 6 factors, and these are the microflora NGS variables, so the diversity indexes, etc. Okay, let's see if there is a significance in this experimental design. So let's go in 50/50 MANOVA, load X and Y, read file, microflora NGS, open, check, always check if this is right. You have to consider if this is the right number of rows and column, because sometimes we had other things within the sheet and we have to erase everything. Leave only the sheet with the data. So the first columns 1, 2, 3, 4, 5, or whatever are the factors expressed as 1, 2, 3 numbers, and the rest are the quantitative variables. Of course, the labels of the first row should be different for each column. Okay. In that case, the columns are the first 6. Okay. And for the X design, so the factors. And what about the quantitative variables? They are many, so we can also do it in that way. We select all and unselect the first 6. 1, 2, 3, 4, 5, 6. This is a trick. Okay. The model. For example, let's try to observe also the interactions. So select all. Okay. Run. And we have to observe, sorry. We can copy in, okay. Okay. So the interactions, there are too few observation to conduct interactions, of course. And so we have only to observe the single factors. So environmental zone is not dependent, while partner code is highly significant. So there is a strict significance to partner code with respect to microflora NGS. And also treatment is not significant, but condition is highly significant. Management is not significant, while crop code is very light significant. Okay, so let's try to observe which variables which of the variables give the higher contribute for partner code, and then we will try for condition. So rotation test, partner code, run. Okay, let's see. We have only the first two have a highly significant contribute. So richness ITS and the faith PD ITS. I don't really know what does it mean, but maybe some of you, I don't know if Francesco is here or also Loredana, I don't know if they want to give a few words on the meaning of this significance given to these two variables. Are you there, Francesco or Loredana? Maybe not. Okay. Or someone of you, if you want to give a meaning on this fact. Okay. You are too shy for me. Okay. So let's go ahead. We can close this and try to observe considering the other that was the condition. So let's see the condition, run. Okay. Here we have more than two. The first five, these first five are significant, highly significant, but also Shannon 18S is significant. So, but the most important are Faith PD ITS and Shannon ITS. Maybe the same, I can't remember, but it's maybe the same of the other analysis. That's it. I suppose that we can stop here if there are no other questions or something that you want to share. Otherwise, we finish right now. Okay. Thank you, and see you tomorrow.
Sorry.
Thank you, Victoria. Yes.
Thank you, Corrado. I have a question about the registration of yesterday.
Okay.
I tried to see download or at least access to it, but it asked me the password to CREA.
Okay.
So it's not possible to see again if I would like to.
I don't know if Loredana is still here, but she downloaded the lesson of yesterday, and she will put on I don't know where, but Exertis will share these 3 days of presentation. So maybe not today or tomorrow, but maybe next week you will be able to look somewhere. They will give a link to view again my lessons. Okay, Andrea?
Thank you.
Okay. Alessandra, maybe you had another question?
No, sorry. It was clear to me. I only want to thank you again for your support. I had the same problem with the other colleague, so I have to check with the Loredana if we can solve the problem for CREA.
Okay. I yesterday downloaded the file, but Loredana told me that she collected all the video and sent to Exertis. So from Exertis, they will share in some way in a web page maybe the 3 lessons. So next week you will able to link the 3 lessons.
Thank you so much. And I have only to try for MANOVA software because I have a wrong message when installing it, and I have to verify if it is a problem of download, perhaps not to get too fast. So I have to verify, to disinstall the previous download and restart again. Because
does anybody of you had problems with installing, or does anybody installed correctly, properly, or not? Any feedback from you?
No, all okay.
It works. Okay.
It works.
So for any problem, Alessandra, you can contact me. Maybe from next week. My support is always available. Don't worry.
Thank you so much. Thank you so much.
Okay. So have a nice afternoon and see you tomorrow with nonlinear approaches. Bye-bye.
See you tomorrow. Bye-bye. Thank you.
Thank you, Corrado.
Thank you.
Bye.
Thank you.
Bye, Corrado.
Bye.
Bye.
Bye.